Hello everyone, welcome back to my channel. Today I'll be discussing about the process of meiosis. In my previous video I have discussed about the process of mitosis, and if you have not seen it yet, please do watch that. And today also at the end of the video I'll attach a question. Do not forget to answer that. So let's start with today's video. We know that the cell cycle comprises of interphase and M phase, and in the M phase either mitosis will take place or meiosis will take place. And in my previous video I have discussed about mitosis, and today I'll be discussing about meiosis. So now let's see what actually the process of meiosis is. So here we can see that this is the outline of the process of meiosis. This is the parent cell having diploid number of chromosome, and in meiosis the cell will undergo two divisions. At first meiosis 1 will take place, and then meiosis 2 will take place. And in meiosis 1, when the nucleus division take place, the number of chromosome will get reduced to half. See here in the parent cell there are 2n number of chromosome, and here it got reduced to n. This is called karyokinesis 1. And after the first nuclear division, the cytoplasm will divide, and then at the end two daughter cells will be produced, and each of them will have n number of chromosomes, half the number of the chromosomes as that of the parent. So since the number of chromosomes get reduced to half, this process is also known as reductional division. And now after the end of meiosis 1, when two daughter cells will be produced, each of the daughter cell will enter into interkinases. Now we have seen that in mitosis, after the formation of the daughter cells, each of the cell will enter into interphase where they grow, and the amount of the DNA will gets duplicated. But then in interkinases the DNA duplication will not take place. The cell will rest for some time, it will gather the materials which will be utilized in the subsequent phases, and then it will enter into meiosis 2. And in the meiosis 2, each of the daughter cell produced in the meiosis 1 will divide again. At first the nucleus will divide, and then the cytoplasm will divide, and as a result 4 daughter cells will be produced. 4 daughter cells will be produced, and each of them will have half the number of chromosome as that of the parent cell. So now where does this process of meiosis take place? The process of meiosis takes place in the reproductive cells of an organism, or we can also say in the germ cells of an organism. So now what is the significance of meiosis process? After meiosis, gametes are formed, and these gametes will then assist in the sexual reproduction process. And in meiosis there is a process called crossing over, and by that genetic informations get exchanged, and as a result variation will take place. And so the siblings are not identical to each other or to the parents. And it also maintains the constant number of chromosomes in a species. Like in human being we know there are 46 number of chromosomes. But then if meiosis did not take place and the chromosomes number did not get reduced to half, after each fertilization the number of chromosomes will get doubled. And then the number of chromosomes, the 46 number of chromosomes will not be maintained in the Homo sapiens species. So it is very much essential to maintain the number of chromosomes in a species. So this is the outline of the different phases of meiosis. At first meiosis 1 will take place, and in this karyokinesis 1 will take place first. The karyokinesis 1 is divided into prophase 1, metaphase 1, anaphase 1 and telophase 1. Now we can remember the sequence by PMET. Now in meiosis 1, the prophase 1 is further divided into 5 phases. These are leptotene, zygotene, pectine, diplotene and dikinesis. Now how to remember this sequence? You can remember it by Lalita, Zara, Pani, Dede. So now there are 2 Ds. So we can see that all the teen will come together, and at the end dikinesis will come. Now after prophase 1, metaphase 1 will take place, anaphase 1 and then telophase 1. After this the cytoplasm will divide by cytokinesis 1, and 2 daughter cells will be produced. The 2 daughter cell will enter into interkinases, and then meiosis 2 will take place. And in meiosis 2, first karyokinesis 2 will take place, where it is divided into prophase 2, and here it will not get divided into the further phases. There is no leptotene and all. So it is prophase 2, and then metaphase 2, anaphase 2, telophase 2 and then the cytoplasm will divide by cytokinesis 2, and as a result 4 daughter cells will be produced. So now as the cell enters into the leptotin stage of prophase 1, we will see that the chromatin networks will begin to condense, and it will condense in such a way that it appears like a beaded structure. Beads will appear, and now these chromosomes will remain attached to the nuclear envelope at least at one end, and it will give an appearance of a bouquet. And hence this stage is also known as the leptotin stage, is also known as bouquet stage. The nuclear membrane, the nucleolus, will not disintegrate yet, and the centriole will also not start moving towards the opposite pole. So this is what happens in the leptotin stage. So now as the cell enters into the zygotin stage of prophase 1, the pairing of the homologous chromosome will take place. So now what are homologous chromosomes? So we know that in human being there are 46 chromosomes, or we also say 23 pairs of chromosomes. Why do we say 23 pairs? This is because one set of 23 chromosomes we get it from the mother, and the other 23 chromosomes we get it from the father. So what happens for any characteristics, like for the eye color or the hair color or the height, we have two alleles. Like if these two are the homologous chromosomes, then they are similar and the DNA sequence in them are also similar. So if this portion of the DNA sequence code for the height, then the corresponding portion of the DNA sequence in its homologous chromosome will also code for the height characteristics. So this may be small t, or capital T, or small t, capital T. This will vary. So now these are the homologous chromosomes. They will come together and they will form a pair. This is called synapse. And then this is called synapse, and then to hold the pair together, there is a complex called synaptonymal complex to hold these homologous chromosomes together. And now here we can see that there is only a single strand of chromatid, but is it actually this? No. This is actually, there are actually two sister chromatids in each chromosome. So we can also draw it like this, and if we draw it like this, we can also call it a tetrad or a bivalent chromosome. Tetrad because it has 4 chromatids, and bivalent chromosome because there is 2 chromosomes. So now as the cell enters into the Pachytene stage of prophase 1, we can see that crossing over takes place. Now what happens? These 2 red chromatids are the sister chromatids, and these 2 green chromatids are the sister chromatids. But this red chromatid and green chromatid are non-sister chromatids, and crossing over will always take place between 2 non-sister chromatids. Now the point at which the crossing over will take place is called chiasma. And to hold the pair together, synaptonymal complex is still there. And here we can see that the nuclear envelope nucleolus did not disappear yet, and the centriole is still in its position. So this is what happens in the Pachytene stage. So now as the cell enter into the diplotin stage of the prophase 1, the exchange of the genetic material will take place at the point where crossing over took place. And now the synaptonymal complex will begin to break, and because of which the terminalization process will begin. And what is this terminalization process? The point where the 2 chromosomes, where the 2 chromatids were attached at the chiasma, now this will start moving in a zip-like fashion, and it will reach the tip, and then it will get separated. So now the terminalization process will just begin in the diplotin stage. Now it will enter into the diakinesis stage, where the terminalization process will be complete and the chromosomes will be separated, and the centriole will move towards the opposite pole. Spindle fiber will form, and it will attach to the kinetochore of the chromosomes. The nuclear envelope and the nucleolus will disappear. So now with this, the prophase 1 of meiosis 1 will end. And now as the cell enters into the metaphase 1 stage of meiosis 1, the chromosomes get condensed completely, and now the homologous chromosomes will align themselves in the equatorial plane. because of the differential forces that acts here, and we have studied this in my previous video, so they will align in the equatorial plane. So now as the cell enters into the anaphase 1 of meiosis 1, the chromosome of a homologous pair, if this is a pair, then one chromosome of this pair will move towards the opposite pole. Because of the pull by the spindle fiber, the chromosome will move towards the opposite direction, and from this pair, one chromosome will shift to this direction and the one to that direction. And what happened in mitosis is that when there are 4 chromosomes, 4 chromatids of each chromosome will shift towards the opposite pole, and there is a division of the centromere. But then here, division of the centromere is not there. A chromosome of the homologous pair will move towards the opposite direction, and that is why the number of chromosomes get reduced to half. So now in the telophase we can see that nuclear membrane and the nucleolus reappeared again, and here we can see that there are 2 chromosomes and there is also 2 chromosomes, whereas in the beginning we have seen, or we have started with 4 chromosomes. So now as the karyokinesis 1 ends, now the cytoplasm will begin to divide, and at the end we will see 2 daughter cells, and each will have half the number of chromosomes as that of the parent cell, and the nucleus and the chromosomes will be recombinant, that is, there will be variation. So now as the haploid daughter cells produced by meiosis 1 is ready to enter into the meiosis 2, it will first undergo karyokinesis 2, where the nucleus will be divided, and this process is similar to that of the karyokinesis of mitosis, which I have already explained in my previous video. And then the cytoplasm will divide by the process of cytokinesis 2, and at the end 4 daughter cells with half the number of chromosomes as that of the parent will be produced. So with this we have come to an end of today's video. Do not forget to answer the question and do like the video if you have liked it, and share it with your friends. Thank you.